I'd like to tell you about our experience using immersive virtual reality to communicate the potential of CRISPR gene editing to be transformative treatment for sickle cell disease. Our team includes Megan Hochstrasser from the Innovative Genomics Institute at UC Berkeley, myself, Lee Bishop from UC Berkeley's Lawrence Hall of Science, and Laura Lins Gonzalez from the 3D visualization firm Dynamoid. Our main goal is to give the general public a basic understanding of how CRISPR works and what it can do so that people are prepared to consider and discuss how this technology should or shouldn't be used. Through our work, we always try to keep in mind that the general public is largely unaware of CRISPR and its capabilities. In surveys of people who participated in our CRISPR planetarium shows, over a quarter of people had never even heard of CRISPR. So the need is out there to be met. We also wanted to raise awareness around sickle cell disease, disease which in the US primarily impacts Black Americans, a population that continues to be marginalized and mistreated in healthcare. In addition, we want to use this as an opportunity to highlight the critical importance of curiosity-driven basic science research, given that this is how CRISPR was discovered and developed. So what is CRISPR VR? It's a virtual reality experience that transports participants inside the body of a simulated sickle cell patient. They can zoom inside their blood vessels to see the blockage composed of sickle-shaped red blood cells, and zoom inside those cells to see the hemoglobin fibers responsible for deforming the cells. They can even zoom into a cell nucleus to see the genetic mutation that gives rise to the whole disease. When participants are zoomed all the way to this DNA level, they can even see Cas9, the CRISPR protein, cut the DNA chain and set off the cascade of reactions that in the end cures this simulated patient. They can verify the patient was cured by zooming back out to see their healthy red blood cells and their blood vessel blockage removed. One of the most exciting results of this project was seeing the number of different ways we have been able to use it. Of course, we've used the VR experience with individual people, but we've also successfully had one person walk through the VR experience while projecting their view onto a 2D screen for other people to see and discuss. Dynamoid has also converted this VR experience into a planetarium show that is being merged into the Lawrence Hall of Science's regular planetarium offerings. In the planetarium show, entire groups of people can be immersed in the same environment and discussions as they're transported to different environments inside the sickle cell patient. We've also exported the VR experience in a number of different video formats. We've used 360-degree video exports in virtual field trips to the Innovative Genomics Institute labs. We found that simple, flat 2D video exports can be quite useful and versatile. We've used them in slide decks at a variety of different events. We've also used video segments in a formal high school curriculum, CRISPR curriculum unit developed jointly by the Lawrence Hall of Science and the Innovative Genomics Institute. Finally, we're also advising university biology instructors who are incorporating this experience into their classes. We've been really encouraged by our evaluation data so far. At our outreach events, survey respondents say that the VR experience is either their single favorite or one of their favorite parts of our outreach offerings. In surveys of the planetarium show participants, 89% of respondents said they would like to see more shows with similar content. We're also pleased that 93% of parents felt that the content was appropriate for their children who ranged from 4th grade all the way down to kindergarten. We feel confident that one of the main drivers of this very advanced content being accessible to such a wide age range is the 3D immersive nature of this experience. Indeed, a growing body of research suggests that compared to learning using 2D visual artifacts, VR can increase understanding and other key learning factors like engagement and motivation. In the future, we'd like to compare learning outcomes directly between the 2D video exports and 3D immersive experiences. We would also like to see how this immersive experience can be paired with additional learning modalities like hands-on activities. Finally, we're exploring the potential of using CRISPR VR as a focal point for engagement with the sickle cell community. We're hoping it can be a resource to them and that we can collaboratively iterate on the experience and perhaps go some way toward rebuilding trust between scientists and people living with sickle cell disease. So we have a lot left to explore and are very optimistic about the future of this project.